he works with Oliver Jensen and also Dr. Horsfield, and also with Jim Wild, Professor Jim Wild, whom we also collaborate with. His background is in physics. He's been working on respiratory modeling, particularly gas mixing, for the last couple of years maybe.
Yeah.
He's here for like 3 to 4 weeks, and we hope to further extend his collaboration and try to find out common interests. So.
Brilliant, thank you. Yeah, thanks for inviting me, well, letting me speak while I'm here. I'm here for another week, so if you have any questions or if you want to chat, then I'll be happy to meet with anyone. As I said, so I'm from the University of Manchester doing a postdoc. My PhD in a sort of former life was in soft matter physics, but this position, fellowship now started in November 2017. Okay, so I'm looking at models of ventilation heterogeneity, and in particular focusing on inert gas washout. So I'll tell you about what those things are if you don't already know. And the question I'm kind of posing here is, how much detail do we need to go into with our models to capture what we see in real data? This doesn't work. Okay, so first I'll just give you a brief introduction to what the inert gas washout and ventilation heterogeneity means. Then we're going to look at trying to infer from real data what's going on in the lung, essentially, how much ventilation heterogeneity we're seeing. Next we're going to start looking at more detailed models, so patient-specific airway models, and trying to reduce the complexity of these down so we can use them in inverse problems. These 2 points are very much work in progress, so none of that's published yet, so you'll have to sort of bear with me on that. And then the final stage, which was actually the work I did first, is all about estimating uncertainty in these reduced models, and that is published, so I can point you to a source for that one. Okay, so ventilation heterogeneity, if you're not familiar with the term, then the idea is that in various lung conditions, what happens is that small regions in the lung become underventilated due to various causes. So for example, in cystic fibrosis, you get mucus plugging the airways and also inflammation, narrowing the inside of the airways. In asthma as well, inflammation and widespread constrictions. And the effect of this is that less gas reaches certain pockets of the lung. And if you look at, say, a hyperpolarized helium MRI image of where the gas is going to in the lung, you see lots of little dark spots. This is a cystic fibrosis example in the figure here. Normally, you would like to see a nice, evenly ventilated lung with the same amount of gas reaching all the lung units so that you have optimum lung function. And this is as much a sort of symptom of other things happening as it is a problem in itself, because you can cope with a certain amount of ventilation heterogeneity and not really notice that you're not really have any downstream effects on perfusion or anything that noticeable. So we're mostly interested in cystic fibrosis with the data sets I'm working on. So if you're unfamiliar with cystic fibrosis, a brief overview here. So it's a genetic condition. It's the most common life-limiting genetic condition in the world, I think. About 1 in 3,000 newborns are born with the faulty gene, which is the CFTR gene. And that regulates ion transport across cell membranes. And when that's faulty, what that means is that essentially the mucus that is normally in many ducts of different parts of the body, but in particular in the lung, the mucus lining is much stickier, it's not well hydrated, and the fluid layer underneath the lining is also dehydrated. And so the cilia that normally push the mucus out of the lung in a kind of conveyor belt mechanism, which captures pathogens and then it removes them from the lung, that can't function normally. The mucus builds up, and instead of being a defense against pathogens, they actually become a sort of really nice environment in terms of for the bacteria to live in and colonate, and you find it really difficult to remove infections once they they sort of take hold. But from a kind of lung perspective, then the first effects you see are essentially this increase in ventilation heterogeneity. Where the airways are filled with mucus and there are constrictions, you get less gas reaching those parts of the lung. And these people will have normal spirometry, especially young children. They'll have normal spirometry results and not feel short of breath or anything, but it's a symptom that things are happening in the small airways that will later have worse knock-on effects and lead to more permanent damage. So this is where this multiple breath washout or inert gas washout method comes in. It measures the ventilation heterogeneity much more sensitively than spirometry, and therefore you can sort of know when changes are occurring before you'd see them in spirometry or even in CT. So an overview of the test, you have some mouthpiece that the subject breathes through, and you start with it connected to some bag of air with a small concentration of whatever your test gas is. In this case, it's SF6. And you wash in, breathing in and out from that bag until your lung is at the same concentration as the bag. So you can be fairly confident you've got a roughly homogeneous distribution of this gas in your lung. And then you switch the source back to fresh air and trace the flow rate and the concentration at the mouth. So I don't know why I'm not using this, the flow rate and the concentration at the mouth. And essentially, the longer or the more gas you have to turn over using regular tidal breathing over the course of that test to clear the inert gas you breathed in is essentially a measure of how well ventilated your lung is and how heterogeneous the ventilation is. So an index that's derived from that, that's most common one used in research is the lung clearance index. So the idea is you integrate over all the flow that's expired, and then this gives you your total flow expired in litres, say, and then you estimate the FRC volume of the lung by integrating essentially the total inert gas that's been expired, because you think the lung was roughly evenly filled with it before, and divide that by the concentration, and then you get you take the total gas exhaled, divide it by the FRC, and you get a normalized measure. So how many times would you have to turn over your lung volume to clear this inert gas down to 2.5% is the arbitrary cutoff point used, essentially. Turns out to be quite good, but originally it was an arbitrary limit set. And so it's a unitless measure. Average adult values come in at sort of 6 to 7, and anything higher than that fairly robustly indicates that there's some ventilation heterogeneity present in the lung. Another measure that's used less commonly, but I'll mention briefly, is phase 3 slope analysis. So if you look at a single breath of that washout, then it'll look something like this. The gas concentration starts off at 0 as you're clearing your dead space, and then goes up to some end tidal value. And this slope here is meant to be indicative of essentially indicative of heterogeneity in the lung again. But to reduce that down to individual indices, you have to plot these for each breath. And one particular measure called S-cond is essentially the slope of the slopes, essentially, how the slope increases with each breath. And another measure, essentially, if you take that linear slope, put it through the origin and subtract it from the original ones, the other index often used is S-asin, which is the first breath, essentially, slope. The idea is that there are two different mechanisms involved here. So S-cond is meant to be the conducting airway asymmetry. So if you imagine that diagram the MRI image I showed you before, where the ventilation is patchy, that never evens out during the test because the lung units aren't really that well connected. They're only connected by conducting airways. So each breath, the difference in concentration in those lung regions are getting more and more different. So each time this slope, which kind of measures the heterogeneity, is getting bigger and bigger and bigger. Whereas the original slope, the idea is this is caused by the structure of the acinus itself, and at the end of each breath, they're relatively well mixed. So the idea is that kind of that contribution kind of resets every breath and doesn't build up and get worse and worse. Obviously, it's not as clean cut in that in real life, but there's a bunch of models, including a few from the ABI, which kind of explain this in a mechanistic way, a bit better than I have done in a minute there. And a sort of newer and more state-of-the-art technique of measuring ventilation heterogeneity is the thing I already showed you, which is hyperpolarized gas MRI. The idea of this is you breathe in some gas that essentially shows up on the MRI machine, in this case hyperpolarized helium, and at the end of each breath you image the concentration of gas in the lungs. You get a spatial picture of how much gas is distributed in the lung, and then you stack those images for a few breaths and you get a kind of decay, a measure of the decay in each voxel essentially of the lung, and that kind of tells you how each individual lung unit is washing out the gas. And regions where there's pockets of poorly ventilated parts, you can see where they are and just how big they are, and then estimate things like how much of the lung is not working properly, things like that. So that's really beautiful data to be able to work with, but also you can't collect it on a large number of people because it's expensive and not many groups have the equipment to do it. So the top one here is what you'd expect to see normally. Some differences due to gravity and things, and differences in tissue compliance and things like that, but it's relatively smooth. And this is a CF patient with this patchy distribution you see again. And if you kind of plot a histogram, you can see it's a bit broader as well. So that's just a histogram of this intensity map.
Is that heterogeneity partly due to the low density of helium? So, for instance, you've got the washout being or lower fractional ventilation at the top of the lung, or are these people lying down?
Yeah, these people lying down. Yeah, so I don't think that has much of an effect. There is a gravitational gradient you can see in the other plane, in the other direction, but I guess it's hard to disentangle what's caused by the fact that the gas is lighter than air and what's caused by the differences in the tissue mechanics, I suppose. Okay, so we started looking at this data from a fairly simplistic point of view. Can we plug some of the MRI? Our first question was, can we plug some of the MRI data into a compartmental model and then run a simulation of the multi-breath washout and see if the two agree? Because and we have a data set where children aged up to 16 with 9 healthy volunteers, 12 with mild CF, so that means they have normal spirometry and CT, and in most cases normal lung function generally as well. And the important thing was the multi-breath washout test in this case was done in a supine position, which is unusual, so that it could be compared directly with the MRI, which is also done supine. So yeah, the idea was to stick a model in between these two essentially and try and simulate the multi-breath washout first from the MRI. Okay, so the model we tried first was essentially the same model people were looking at back in the 60s and 70s. It's essentially a collection of bags modeling the lung units and connected to the mouth by independently by tubes which represent the airways or the dead space. And the inputs you put in to the model are the tidal volumes, the dead space, and the distribution of ventilation, so how much gas is going to each bag, the total volume, and also because this makes a small difference to the measured LCI, and it's taken into account in some models, the re-inspired gas. So you can re-inspire some gas, and then that kind of slows down the washout as well, so you account for that. So yeah, these kind of models, this one's slightly different, but taken for sort of PNAS 1963 was the first one to try and fit this kind of model to some data. But they didn't have the MRI technique to compare to back then, or to put in. So in a given lung unit, we define some parameter x, which is essentially the tidal volume, or the amount of gas going to that unit, divided by the mean of what goes to each tidal unit on average. So if x is 1, then it's a perfectly average unit, and x less than 1 means underventilated, x more than 1 means ventilated more than the mean. And the concentration, this gamma, in each unit at the end of breath n is essentially proportional to the concentration at the end of the last breath. And then there's a dilution factor, which is basically proportional to the volume before inspiration divided by the volume after inspiration, but taking off the contributions of like re-inspired gas and things like that. And if they don't clear the dead space, then you don't want this term going more than 1, so there's a maximum cap of 1. The concentration can't go up as the test goes on. And this delta V, which is the volume the volume change in each lung unit between breaths, tracks if they have a particularly uneven breathing pattern, which some people do. This model was tracking the changes in tidal volume to keep account of that. Okay. So then the concentration that comes out of the mouth at the end of the breath in this model is essentially just a weighted integral of all the contributions from each individual unit. So we take the continuous limit, and px here is just the probability distribution of the different ventilations. It's weighted by x because the more well ventilated a unit is, the more volume it expires, and therefore the more it contributes to that to what you see in the signal at the mouth. And gamma is just the concentration being expired at the end of the breath. And we put in there's a minimum limit. If it doesn't clear the dead space, basically you don't see it at all on the signal because that unit's so poorly ventilated that it doesn't contribute to the model. Okay, so this is just to show that even though, so this was choosing a simple form for the distribution, which is just 2 parameters. If we put in real tidal volume data, which is these blue and black dots, which even though people are told to try and keep their breathing, regular, you try and breathe half a litre every breath, it's really hard to keep reproducing it. It does vary a bit. And so the end tidal concentrations can move around a bit just because of that, essentially, in the model, even though there's not an extra parameter for that. So the red line, the concentration here, the idea is if it's moving around just because of changes in tidal volume, the model should be able to account for that, even though it's pretty simplistic. Okay, so we stuck the MRIs from this data set into the model with dead spaces and FRCs taken straight from the multi-breath washout data. So the only thing we're testing really here is if we put the heterogeneity we observe into the model as that distribution P of x that I showed you, then can we explain the multi-breath washout data? And in this data set, the answer is not really overall. So here is, so orange is CF patient, blue is healthy volunteer. We see fairly clustered good agreement when the ventilation heterogeneity, or the lung function, is normal essentially, which is this 6 to 7 window of LCI here. There's 4 cases that are measured to have increased LCI, but from this simple model, putting in the MRI data, they come out essentially healthy from the simulations. So there's something missing, whether it be in the data or in the model is still a slightly open question. There's something that's not being explained that we can see in the LCI and we can't see in the MRI data. Some potential explanations, which I'll talk about in a minute. There's one case that agrees really nicely. So if you look at the Bland-Altman plot, the agreement limits are quite poor, plus or minus 2 on LCI, which when your window of healthy is about 1 and a bit wide, then that's not great. Okay, so I've produced that first because that it makes logical sense to do that first, but that's not actually what we did first. What we did first was try to fit the model to the data, and this was by way of an explanation. So we did look at an inverse model case to see, okay, if we go backwards from the multi-breath washout data, how similar does it look to the MRI data? So we have to choose a form, or you don't have to, but it's a very ill-posed problem to try and infer a continuous distribution from essentially a zero-dimensional signal at the mouth. So we chose a form of the distribution that's often used in the literature. We take a log normal distribution and we chop the ends off it because we don't want it to go to infinity, because we want to simulate the thing. So we have to impose volume and flow conservation. So the mean ventilation has to equal the mean ventilation we observe. And so that brings these two conditions onto this, which means it's basically fully defined from one parameter in this case, which is this sigma, which is the width of the distribution. So there's only one parameter describing this distribution in the end, because the mean is fixed by the volume and flow conservation. Okay, so we fit the FRC volume and sigma. We could also have tried fitting the dead space, but for that, the Fowler dead space we measure is so consistent in the multi-breath washout that we'd essentially end up with the same result as just putting it in the first place. So we just stuck the Fowler dead space into the model as the model dead space. So that's that. And we stick in the measured tidal volumes as well. We correct for things like BTVS and also making sure that by the end of the test, the inspired and expired volumes come out the same. So we haven't lost lung volume, because there are factors that mean you do lose volume over the course of the test, including the different density of the gas on inspiration and expiration, and also gas exchange. So we don't model any of that, so we just put in a fudge factor basically to correct for that. And we fit this model using a standard nonlinear least squares method. We estimate the error in the measured data from looking at the variation in the curve near to the end tidal point. And we start by taking the FRC estimate from the raw case, and we just randomly choose an initial sigma, and we repeat until we get them in the same solution found 10 times, essentially. And then we assume that's the global minimum. Because it's only two parameters, we very solidly get the same global minimum out every time. So here's some example of what the fits look like. The dots are the end tidal concentrations. The error bars are estimated error. You'll see a couple of them have really big error bars, and that's because they didn't take a big enough breath out. So the slope was still really large at the end of the breath. And you'll see the healthy volunteers here have really nice straight line washouts on a log-log plot, and we can fit the data really well. The CF patients, this one in particular, I picked this out because it's a particularly noisy example where randomly the end tidal concentration just jumps up halfway through washout, and I don't know what happened there. I guess maybe some lung region that was completely blocked got unblocked somehow, maybe shifted position, maybe coughed something out. I have no idea, but yeah. So just to show that in most cases we get really good fits for the model, but in a few cases you can't hope to explain it with a two variable model. And results agree really well with the multi-breath washout indices. So the fitted FRC really closely fits with what's estimated from the software they normally use to analyze the washout data. And the LCI really strongly correlates with the fitted sigma of this distribution. And if we look at on a patient-by-patient basis, we see we get the same separation of the data as we do in the LCI as well. So if we draw limits, plus or minus, confidence limits around the healthy volunteer data, we can see we get the same separation of those four abnormal ones that we saw before for both either measure. So it seems about as good a measure, at least, as LCI. So I guess the question is it telling us anything about anything extra than LCI? Because LCI is a number, and it's just a number, and this sigma ideally should be telling us about the ventilation heterogeneity that's going on. Some example distributions versus fitted distributions, where all the means are set to 1. So in summary, again, we kind of see what we saw in the forward model. We get good fits, so we reproduce essentially the problem we had before, which is that 3 people we predict should have really broad ventilation distributions, and they don't. And again, agreement limits because of that are quite poor. So why is this? One of the reasons that we think is because this hasn't been done in kids before, basically. This kind of model hasn't been looked at before. And I think the problem is that the tidal volumes during the multi-breath washout test are quite small. You're talking 300, 400 mils. And the MRI test, to get a good signal, they use a litre bag. So generally the tidal volume is around a litre. And these kids' lungs are only up to 2 litres at most. So you're breathing in quite deep breaths, and I suspect the ventilation profile is going to look quite differently when you're doing a kind of almost forced inspiration compared to this relaxed breathing that's in the MRI test. So we think that might be one thing that's going on. So we probably need a better model than we have, because our model at the moment essentially just maps linearly from one state to the other, and it's almost certainly not a linear process going from tidal to forced breathing, essentially. The other thing is that it might be below, some of the heterogeneity might be below the resolution of the MRI. We know it happens in the small airways. If it's the really small airways, then it might just be averaged over by the imaging, and so it looks smoother than it actually is. The other point is this model is basically the simplest model we could have fit to the problem. So perhaps we need more detail of the actual physiology in there to explain, to see what's going on, because there's clearly a signal in the multi-breath washout data. And if you look at the MRI data, you can see patchier distribution, patchier ventilation, but if you look at the overall lung distribution, it doesn't look too different. So maybe we're missing some details by taking too crude a model. So that's what we're working on now, essentially, as well as maybe adding more data to that and seeing if it works better in adults, for example. So very briefly, I'm going to talk about what I'm working on right now, which is similar to what Amir, Merin's PhD student, has also been working on as well, which is taking patient-specific airway tree models and essentially averaging them down to be computationally more efficient so that we could potentially use them in inverse models like before. So if you do a full ventilation transport simulation on a full patient-specific airway tree, 30,000 terminal airways, then you're talking, maybe at best maybe like 20 minutes, half an hour per simulation. And then if you want to do 1,000 of those, then you're going to very quickly sort of run into problems without using a large computer. Okay, so so that's essentially the problem. So I guess the question is, do we need all those airways, or can we sum them up by chopping parts of the tree off? So what we do is we average essentially over the more distal airways. We turn those into symmetric trees, which can then be represented by a single edge or a single line of nodes rather than lots and lots of branches. So if you imagine if we approximate the structure as a symmetric tree, then you can represent this symmetric tree like every single airway in a generation is going to have the same properties, so you can just represent them all by one representative edge and take account of where all the volume goes. So to do that, we choose to try to retain essentially the functional properties of those subtrees that we're going to replace. So we measure the resistance of the subtree that we're going to replace, and we measure its total volume, and then we replace it with a symmetric tree that has those same properties. And also as close as we can get to the same number of terminal branches as well. So that defines our radius and our length of our branching tree. And then there's just a relation between the size of each generation. So if we do that, then for example, here's a full patient-specific 1D airway centerline data with branching algorithm to fill the rest of the volume. Probably seen quite a few of these, I imagine. And this is an example simulation, a snapshot of a simulation where there's some fairly heterogeneous distribution of concentration that's been transported around the lung. And if we go up to Horsfield order 8, so this is 8 generations up from the bottom of the tree, and chop there and reduce down to symmetric regions, then you get 10 times reduction in system size. Everything looks pretty similar in terms of the concentration, and then you can keep going and chop down to something that doesn't look that pretty anymore, but still captures some of what's going on. There's obviously going to be a point where you just lose too much information and your simulations become no better than a really crude trumpet model, essentially. So there's a cutoff somewhere to be found. So we're looking at this cutoff in a case where there's a slightly elevated LCI, and as you average more and more generations, your LCI drops because you're kind of averaging everything over. The ventilation heterogeneity goes down, and the error, which is this orange line, the relative error from the baseline position, from the baseline washout curve, just goes up and then very sharply increases as you get to essentially replacing all the branches with something symmetric. And we also tried, instead of going from counting from generations from the bottom, if you count generations from the top, does that give you a better averaging procedure? And the answer is no. It always seems to be better to go from the bottom up. You seem to retain more asymmetry in your structure and stay closer to the original structure of the tree. So the relative error is always less for the same saving in number of airways. So this is still essentially ongoing. You can see that the red, blue, and black lines are essentially, the black line is the full simulation, the blue line is when we chopped 8 generations off, and the red line is when we chopped 16 generations off, and this is the first breath of washout and the last breath of washout simulated. And you see very little change. But one thing we can do, once we've chopped some generations off the tree, we can put a more complicated Asmus model in as well, because we've made some computational savings, so we can put a detailed Asmus model in, and that's this green line, which shows that can make quite a big difference to the simulation. So by making savings in one place, we can add complications in another place as well, and still hopefully retain that patient specificity, which is a cool thing to have in the simulations. Next question is, I guess this was all starting with a linear model, so how when we simulate more complicated things like nonlinear resistance and nonlinear compliance and things like that, how well do they average down, or do we lose information more quickly in those cases? And are there smarter choices for which regions to average? This is something I've been doing in the last couple of days, actually, is if you just look at the pressure drop over the whole system, simulate pressure drop over the whole system and look at the flow out at the ends, maybe you can just you can see regions that are more homogeneously ventilated and average those out, and then leave more heterogeneous ones alone. And also, we could also trim down models of the Asmus in the same way, is the idea. And the final question there is, can we estimate how much is lost by making these approximations? Obviously, we showed before how much information we're losing in a case-by-case basis, but that involves simulating the full case every time, and ideally we don't want to do that in the end. So this ties back to the work I actually did first, chronologically speaking, which was a perturbative method, a model of heterogeneity. So this is looking at starting with those symmetric trees, where all the branching is the same in every direction. So this is an example model. You have some asymmetry in the top few airways. This is a schematic diagram of the airways, and then at some point we just cut off and replace with these symmetric single edges that represent the rest of the tree. And if we want to simulate constrictions, then we have to maybe go down to a bit more detail and cut off later on, because if one airway is severely constricted, then you don't want to average over that. So this was the simulation setup, and the idea is you compute the flow for the ventilation, update the gas distribution through a transport equation, and repeat. And all of these tree models assume some kind of simplistic scaling relation, so that we can build it without, these models were all built without any patient-specific information to start out with, so it was models taken from the literature, essentially like the adapted from the Horsfield and Weibull data. So the equations for the flow will be very familiar. Pressure drop over the airways is proportional to resistance times the flow. We just use Poiseuille resistance in this case. And the sum, if you look at a single node, the sum over all its connected edges of the flow has got to be 0. That's just incompressible flow. So this is in network notation. So the edge ij connects node I and j. There's a linear viscoelastic assumption for the Asner units at the end, so they just expand with some elastance and resistance, and there's a driving pressure term, the pleural pressure, which is assumed the same for all the units. And then we also simulate flow in the Asner ducts, but they are also taken to be symmetric, and so the expansion of those is assumed to be uniform for each Asner. So each alveoli in a given Asner expands by the same amount, and they're distributed according to the surface area of the ducts. And then transport, finally, is just a simple advection dispersion equation you can use. So instead of just molecular diffusion here, you'll have some kind of Taylor dispersion type term to account for that. So that's flow dependent. So first we just played with the model, applied some constrictions to the airways, and looked at what happened to the LCI, the S-cond, and the measured FRC volume as we increased constriction strength. Here we took this region and just constrained it at different depths in this symmetric model, and here we distributed defects randomly onto this network and looked at the change. And what you see is that when the defects are distributed randomly and they get more distal, you see slightly less effect on the multibreath washout indices, but overall it's pretty hard to tell the difference essentially between whether the defects are more distal or more proximal in this model, because essentially at the end they have the same effect. Essentially there's a very broad peak of both of these indices, which are actually quite tight constrictions. So you really have to increase the resistance of these airways quite a lot to get some ventilation heterogeneity. And it's a pretty strong nonlinear response as well. So if you completely close it off, it goes back to normal because you don't see that lung region again. Okay, and this is the simulation video. If you constrict a region, then it also lags behind the rest of the lung because it has increased resistance. You can't really see it so well on this one, but the defects in this case are all distributed around. So what we do is we say, well, if we've already applied our strong constrictions, can we take into account what our how much uncertainty is involved in our averaging? So if we've got this symmetric structure, can we estimate how much we've lost by assuming everything is symmetric without actually just simulating a full asymmetric system? So what we do is we perturb individual airways. So this parameter epsilon is assumed small, and we change the area or the length of a given airway in a simulation and simulate just the linear response in all the properties of the model, so the velocity or the gas concentration, and track those. So we don't actually have to make the model any bigger to do that, because the airways all start out the same size in a given generation. But by measuring that essentially for each airway in the model, we can get a measure of a bunch of linear sensitivities. So you say, if I perturb this airway, how's the system going to change? If I perturb this one, how's the system going to change? So it means you have lots and lots of models all running at the same time, but they're all quite efficient. They're all quite small. And then because you've linearized everything, the idea is Oh, yeah, so this is how you can also try and look in more detail at what would go on spatially by separating out and perturbing a single airway, for example, like this. But I won't go into any detail on that. But the idea is if you have a linear response for each airway, and then what you can do is you can say, well, what happens if I have now some randomly distributed perturbations everywhere? Instead of having to build the full model and simulate, you can do some pen and paper maths, basically, and say, right, I have a normal distribution with, say, 0 mean, because I've simulated the mean case, is the idea, and some covariance matrix describing how all these perturbations are distributed. I can then estimate what the variance of my various measured parameters are going to be by doing basically a big sum of each sensitivity squared, more or less, or well, each sensitivity interacting with each other one that it interacts with via this big covariance matrix. So you're essentially adding up all the variances that come out from each perturbation. And you sort of do that after the simulation has run, and then can try different scenarios with the same set of sensitivities you've measured. Okay, so we also looked at, so we could just have, a Gauss, an independent normal distribution, but we also looked at the effects of correlations as well. And so here is an example where we randomly put independent variations in the radius and length of all the airways, and each cross is a full simulation of the case, and the blue dot is the mean plus or minus some standard deviation, and the red lines are our prediction from our linear theory based on essentially a single simulation. And you see that in the case where there's no constrictions present, our prediction is really good. In the case where there's constrictions to the airways already, then there's a big change in the mean that comes about, because when you start to add randomness on, there's only one way essentially it can go, and that's back down to a slightly more healthier case, because you're already at like the worst case for that part of the lung. And so we estimate the variance still quite well, but we can't track this change in mean because it's a linear model essentially. So all the contributions add up to 0 in the end, because the mean change was set to 0 originally. So it propagates the uncertainty quite well through the model, but it doesn't explain anything more interesting than that, essentially. And the standard deviations you can get standard deviations out of things like the LCI and the S-cond, and you see that essentially your uncertainty is only important when you already have constricted airways in the model already. So if you have some ventilation heterogeneity, there's quite a sensitive response to randomness in the structure, but whereas if everything's healthy and working normally, the system's super robust to small changes in structure, and so your uncertainty just due to some structural heterogeneity is small. So this tells us, I guess, in future, if we were looking at a model that we'd already averaged down in some way, we could get a good estimate of how much uncertainty there was involved in that averaging by just looking at the really underventilated regions. And also from that data, you can try and reconstruct distributions of in this case, fractional ventilation, because in the model originally there were 7 lung units, so your distribution of ventilation was 7 vertical lines, essentially 7 values of fractional ventilation, and by adding in this extra Gaussian distribution on top of them, you can estimate how broad the distribution within each of those regions should be. And the take-home, I guess, is that it's much broader in the region that's already affected by constrictions. So the underventilated regions are much more sensitive, yet again, so that explains, I guess, kind of where this effect is coming from. So you can, so that was published in PLOS ONE, had the link at the start, and I forgot to point at it, but easy enough to find it. So in summary, so the multi-breath washout indices are really useful clinical tool, and they, particularly in early CF in young kids where things like spirometry and CT look perfectly normal, it fills a gap that can hopefully catch changes in lung function really early on. And if we can combine what we know from imaging data with multi-breath washout data, we can hopefully get a better understanding of the early CF physiology, but also develop tools that we can use to analyze the multi-breath washout data and tell us more about the actual physiology. That's the plan. The simple models I've used so far to fit to the data don't seem to explain the data very well. So we probably need to look at more complicated, more complex, more detailed models. And then the work I did first showed how sort of sensitive the multi-breath washout indices are to constrictions, and how it's a really strong nonlinear response. So we essentially, the link between physiology and ventilation is maybe not so straightforward. And so that's maybe why more detailed simulations are required. So I'd just like to acknowledge all the people involved in the work. So my supervisor in maths is Oliver Jensen. My clinician supervisor is Alex Horsley at the Wythenshawe Hospital in Manchester. All the MRI data we used has come from the Polaris group in Sheffield, headed by Jim Wild, and collected by various people, Guillaume, Laurie, Felix, and Helen. And the airway trees that I was averaging, the 2 patient-specific airway trees that I used to test that on, were provided by the Oxford group from the AirProm project a few years ago. And the research is all funded by the MRC in the UK, and initially was kind of, before we got that grant, we had a bit of leftover funding from previous grants that started the whole thing off, so the EPSRC and NHS funding. Okay, so yeah, happy to take any questions, but you're probably all tired now because it's Friday afternoon. Yeah.
Thanks for that, Carl. So you, on the topic of your weak heterogeneity, you mentioned that you examined independent perturbations, and you briefly said you also looked at correlations as well. Specifically, what correlations are you looking at? Is it that there's a greater chance of a parent vessel being perturbed if the daughter vessels?
Exactly that, yeah. So we hypothesis for the putting the correlation. Obviously, you can choose a vast variety of correlation functions, but our idea was that what if structural correlations are inherited? So if the parent branch is slightly larger than average, or the daughter branch is also likely to be larger than average, often you see in the physiology that might be the case, but also, of course, there's asymmetry in the branching as well. So what we put in this case is essentially if the parent is perturbed in one direction, the daughters are perturbed with their parent branch perturbation as the mean. So essentially each one is inherited, is starting from the starting point of that. They only know about their parent branch, they don't know about the whole population is the idea. So they randomly deviate from the parent branch size relative to the population mean. And the difference between these 2 correlated cases is this rho factor, which is the correlation of the area to the length of the branch. So if it's 0, then changes to the radius and the length of the airway were uncorrelated, and if if it's 1, then they were completely correlated. So the area and length, essentially, perturbations were a fixed aspect ratio. Not a massive difference between those 2 cases, but they make a slight difference to how the model responds. Yeah.
This might be a stupid question, but you were talking about the patients that you had, and you had healthy and those with mild cystic fibrosis. Was there any reason why you had ones with only mild?
The main reason, because we were selecting that based on who'd done the multi-breath washout in supine, first of all, because we wanted to not worry about gravitational effects, basically. We didn't want to worry about the fact that they were going from sitting up in one test to lying down in the other. It might be that isn't that big a factor, and we can look at a bigger data. So we wanted to not have that in our first test case, essentially. And it so happened that the data sets that was true for was this mild CF study. Yeah, which was looking at the differences between the two postures originally a few years ago. So we're going to broaden the net. I think it'd be adult CF patients generally have much worse ventilation heterogeneity anyway. So you're always going to get a signal out. So you have to be careful how much you say about that. You're always going to get a nice correlation basically. It would still be interesting if we get good agreement. That would be stronger than a correlation. But yeah, the important thing about the adults is, like I said, they take a similar tidal volume in both tests. So maybe the two situations are actually nearer in that case. And maybe we've got more hope of fitting the model to it, a simple model to it.
Was there any significance to your choice of a log normal sort of distribution?
Mostly that's what everyone's used before, and that's what. So the midget technique, the multi-inert. There's this technique that people. It sounds wrong. It's MIGET. It's Wagner's group in America back in the 80s. They were fitting distributions to the perfusion and the ventilation, and they used log normal distributions essentially because what they observed looked log normal, and you have some constraints. It can't go below 0, so and I guess technically could go up to a very large value. So I guess mathematically it's got very few parameters and obeys those rules, and it does look good for in most cases. If you were to try and fit a normal distribution to the distributions you get out. I can't see if I can see a couple of the histograms that I showed before. They then don't look miles away, but they also look quite similar to normal distributions as well. So, yeah, it's hard to say exactly.
I was just wondering, log normal distributions are often associated with multiplicative sort of processes rather than additive. I don't see any reason why there would be multiplicative sort of processes operating in this system.
No, that is true. I don't know, I guess is the answer to that. Essentially, the main answer is that's what's been looked at before, but that's not a very good answer, obviously. I played around with fitting different distributions to it, and the final results really don't change very much at all. So the choice, especially if you keep it down to one parameter essentially. So I fitted a truncated normal distribution, and essentially the same results come out. So it doesn't seem to matter that much. Even tried more complicated models with more parameters, and doesn't seem to help basically. If that's everything.
Well, let's
Okay.
thank Carl one more time. Carl will be here one more week here, right?
Yeah,
So
that's
we
right.
can continue our discussion. And there are some cookies and apples, so you can feel free to grab it as you go out, or you can have more questions to Carl.
And thank you for coming as well.